Hello and welcome back to Suchitra's Biology. So today we are going to discuss about MHC, that is Major Histocompatibility Complex, and its types. So let's start. MHC, they are present on mammalian species. They are only found in mammalian species and they help us to differentiate between self and non-self, like which cells are our own cells and which is a foreign or antigenic cell. Mammalian species like humans and mice. On humans it is found on chromosome number 6, while in mice it is found in chromosome number 17. Okay. MHC is known as HLA, human leukocyte antigen, while in mice it is known as M2 complex. Sorry, it's mice. Okay. Now MHC is codominantly expressed. It is polymorphic, that is there are various alternative forms of the same gene. Okay. Major histocompatibility complex are tightly linked cluster of genes. So they are very tightly linked. As they are tightly linked, so their recombination frequency is very low, about 0.5% around. Okay. Now MHC does what? MHC is a structure that represents the antigenic peptides, the foreign antigen, to T cells, so that body can recognize that it is a non-self cell and so that the antigen can be killed. Now MHC is of 3 types: MHC 1, 2 and 3. These 2 are very important. This is not that important. These 2 represent or present or show the antigenic peptides to T cells. MHC3 does not show or present the antigenic peptides to T cell. Okay, it does not do that. So now let's discuss MHC1 and MHC2 in detail. First, let's discuss about MHC1. Okay. So MHC1, it is present in all nucleated cells. All nucleated cells means all the cells that come under the category of WBCs, like neutrophils, basophils, monocytes, leukocytes, B cells and T cells. Okay, all nucleated cells. They are present on all nucleated cells. Now MHC1 is made up of alpha and beta 2 microglobulin. Alpha is of 45 kilodalton and beta 2 microglobulin is of 12 kilodalton. Okay. Alpha has alpha 1, alpha 2 and alpha 3. Now let's see the structure of MHC1. This is is alpha 1, alpha 2, alpha 3, and it is beta 2 microglobulin. Okay, this is the plasma membrane. This is the cytosolic end. This is the transmembrane domain. Okay, hydrophobic or transmembrane domain. This. Now alpha 1 and alpha 2. Alpha 1 and alpha 2 form antigen binding domain, or we can say that the antigen peptides bind here. Okay, these are the antigenic peptides. This antigenic peptide binding site that is formed by alpha 1 and alpha 2 is of 8 to 10 amino acids. Okay, this area is of 8 to 10 amino acid. Alpha 3 has transmembrane domain as it has crossed the plasma membrane. But beta 2 microglobulin does not have transmembrane domain. You can see there is no transmembrane domain. Alpha and beta are bound by non-covalent bond. Alpha and beta 2 microglobulin bound by non-covalent bond. Okay. They are bound by non-covalent bond. And MHC1 represents the antigenic peptide to TC cells that have CD8 co-receptor. Let's see how it represents the antigenic peptide to TC cells. Heterotoxic T cells. Okay, it is a type of T cell. Suppose this is unnucleated cell. Any nucleated cell. Virus enters the cell. The virus RNA is made into protein and proteins are negated. Okay. Now there is MHC. It is MHC1. Now these peptides are represented here. Now it is the TC cell. Okay, and it has a receptor, co-receptor, that is CD8. CD8 co-receptor is present on TC cell and MHC1 is present on any nucleated cell. So it will represent to this. So now it can, actually it is closer to that. Okay, so it will now recognize that it is a antigen, and now it will release perforins that will create pore in the antigenic cells and antigen will die. ठीक है, pores बना देते हैं, not in this cell, antigenic cell. Okay, so this is how the MHC1 works. And also each of the alpha has 90 amino acid sequences. Each one. Alpha 1 also has 90 amino acid sequence, alpha 2 also, and alpha 3 also. Okay. Now let's discuss about MHC2. So MHC2 is found only in APCs. That is antigen presenting cells. What are antigen presenting cells? The cells that present the antigen to other cells. Okay, like T cells, APC. APC is like dendritic cell, B cells, macrophages. Okay, these are the three cells that are APC, known as APC. MHC2 is present on these cells, on the surface of these cells. Okay, suppose this is the dendritic B cell or macrophage, MHC2 will be present here. Now MHC2 has alpha 1, alpha 2 and beta 1 and beta 2. Okay, so let's see the structure. This is the plasma membrane. Okay, these are the cytosolic domains. Cytosolic domain. This is the transmembrane domain. Transmembrane domain. Now this is alpha 1, alpha 2, beta 1 and beta 2. Okay. Now in this case, alpha 1 and beta 1 are forming the antigen or antigenic peptide binding site. Okay, binding site, a groove. Now this antigen binding site is where the antigen will bind. Okay, antigenic peptide. It is made up of about 13 to 18 amino acids. This area. Okay, complete. In MHC1, the peptide binding site was made up of around 10 amino acids, but in this it is of larger amino acids. Okay, it is made of larger amino acids. Now this is about MHC2. In MHC2, alpha 2 and beta 2 have transmembrane domains. In MHC1, only alpha 3 had the transmembrane domain. In this case, both alpha 2 and beta 2 are transmembrane. Okay. Alpha and beta are bonded non-covalently. MHC2 represent antigen to T H cells which have a co-receptor called CD4. Okay. So let's see how MHC2 represents the antigenic peptides. This is the APC cell, antigen presenting cell. It can be WBC, dendritic cell or macrophage. Okay. Now there is a bacteria. Suppose this is a bacteria. It's degraded into peptides, bacterial peptides. Now this is the MHC2. Okay, MHC2. This is TH cell having a co-receptor called CD4 co-receptor. Okay, nucleus. Now what happens? These antigenic peptides are represented here. Now the CD4 receptor will recognize these antigenic peptides and it will secrete interferons, cytokines that will attract other cells that will kill the antigenic cells. Okay, this is the part. So this was about the MHCs. Do like, share and comment and do subscribe to my channel.